This is the Bio 104 video for Lab 12, Sex-Linked Genetics, Pedigrees, and Blood Typing. The first part of this lab has to do with the sex chromosomes, and if you remember, every individual has two sex chromosomes, and the two sex chromosomes are X chromosomes and Y chromosomes. The X chromosome is very, very large, and it has many different types of genes on it. The Y chromosome is very, very small and has very few genes on it. And pretty much the genes that are on the Y chromosome are involved in male sexual development. Females are XX, so they would have two copies of the X chromosome. So when you talk about traits where the alleles are found on the X chromosome, females can be homozygous or they can be heterozygous, depending on if they have two of the same alleles on each X chromosome or if they have two different alleles for the trait on two different X chromosomes. Males are XY, and because the X chromosome has the alleles and the Y chromosome has virtually no alleles, males are considered hemizygous. So whatever allele they have on their X chromosome, that is the trait they will express. The Y chromosome, for all intents and purposes, has virtually no alleles. So for X-linked traits, males are hemizygous. So whatever allele they have on the X chromosome is the trait they will express. Often the X-linked traits that we talk about are disorders where the alleles are found on the X chromosome. Some examples of these disorders are color blindness, hemophilia, and muscular dystrophy. When writing the alleles on the X chromosome, there's a certain notation that you have to use. So usually the normal allele is dominant, and so when you write it, you write the X chromosome, so you write the X, and the dominant allele is superscript. So in this case, we're using C, so a capital C would be a normal allele. The disorder allele, again, you write the X chromosome, and now the superscript letter is the lowercase because often these disorders are recessives, so you can underline the C to indicate it is the lowercase. Females. So the genotype for an X-linked disorder in females can be written as XX with two normal alleles. That would be a woman who is homozygous normal. Or if she's heterozygous, you write the two X's with the capital allele and the lowercase allele. Or if she is expressing the disorder, both her X's would have the disorder allele, so lowercase. Males, you write the X and the Y, and only the X has an allele, so only the X has a superscript letter. So this is a male who carries one normal allele, so he has the normal phenotype. This male, the X chromosome, has the recessive allele, so that should be underlined there, has the recessive allele, so he is expressing the disorder. For experiment 1, on your lab report, you're going to do some Punnett squares related to X-linked disorders. And this is an example of a Punnett square with an X-linked condition of eye color in flies. So remember, when doing the Punnett square, the first thing you want to do is figure out the genotypes of the parents. So in this case, the genotypes have been given to you. This is a female's genotype. Female, she has two X chromosomes, and she is heterozygous for the eye color. The male's genotype, he's a male, so he has one X, one Y, and the X has the allele, so in this case he has the red allele, and again the Y does not carry an allele. So you first figure out the genotypes of the parents, and then you determine the different types of gametes that those parents can produce. The female, half of her eggs will have an X chromosome with the dominant allele, half will have the X chromosome with the recessive allele. For the male, half of his sperm will have an X chromosome which contains the dominant allele, and half of his sperm will have the Y chromosome, which has no allele. So once you've figured out the type of gametes that the two parents produce, then you just fill out the Punnett square. So you fill out the Punnett square, you figure out the genotypes, and half of the offspring are going to be female. So half of the offspring will have two X's, and half of the offspring, probability-wise, will be male with an X and a Y. When you're evaluating phenotype, you usually talk about 100% of the females. So this is 100% of the females. So you have two boxes, so each box is worth 50%. So in this case, 50% will be homozygous red and 50% will be heterozygous. Then when you talk about the males, you talk about 100% of the males. And in this case, 50% of the males will have the red I allele, 50% will have the white I allele. So in the Punnett squares, when you're talking about sex-linked genetics, you divide out the males and females. So you're not talking about 100% of the offspring, you're talking about 100% of the females and 100% of the males. The second part of your lab is going to deal with pedigree analysis. Pedigree analysis is a way to follow a trait through a family, and this is usually used to follow the inheritance of a disorder or a disease through the generations of a family. When you are drawing a pedigree analysis, females are depicted as circles, males are depicted as squares, and the affected individuals are shaded. Normal individuals are clear, but the affected individuals are shaded. And for the pedigree analysis, you have to determine the pattern of inheritance of the disorder. And there are 3 patterns of inheritance. Those patterns are X-linked, Autosomal Dominant, and Autosomal Recessive. So this is telling you the characteristic of the allele that causes the disorder. X-linked means the allele is on the X chromosome. So you would write it as XA because it's usually a recessive. Autosomal Dominant means that the allele is on one of the other autosomes, one of the other chromosomes, 1 through 22. So it's a dominant, you would depict it as a capital A. Autosomal recessive means that that allele is on an autosome again, not the X chromosome, and it's a recessive, so it would be a lowercase a. This is a pedigree for an X-linked disorder of hemophilia. And this is a real pedigree for a famous person. This is Queen Victoria of England. And she was actually a carrier of hemophilia. So in this case, the carrier females, the heterozygous females, are denoted by a half-shaded circle. In a pedigree, a marriage is denoted by a horizontal line. So Queen Victoria was married to Prince Albert. And then their children are denoted by a vertical line and then a horizontal line. And Queen Victoria and Prince Albert had 9 children. So these starting with a female daughter, then a male, and going all the way down the line to their last child was a carrier female. For an X-linked disorder, when you are trying to determine the pattern of inheritance, X-linked disorders are found mainly in males. So when you look at this pedigree at the shaded individuals, all of the fully shaded individuals are males. And that is your main indicator that this is X-linked. The half-shaded carrier females do not express the disorder, so they don't quite count for this part. But if you look at a pedigree and the majority of individuals that are affected are males, then it is an X-linked disorder. Another pattern of inheritance that you may come across is an autosomal dominant pattern of inheritance. And with an autosomal dominant, what you will notice is that every affected individual has at least one parent with the disorder. So remember, autosomal dominant, you just need one allele, the dominant allele, and you get the disorder. The best way to evaluate these pedigrees is to start from the lowest generation, the newest generation. So you look at the individuals. So here in this family, these two children, these two siblings have the disorder. You look at the parent, and they have at least one parent who has the disorder. In the next generation, these three individuals have the disorder. And when you look at their parents, they have a parent who has the disorder. So if in every case the affected individual has at least one parent with the disorder, then it is an autosomal dominant. The third pattern of inheritance is autosomal recessive. So this is a disorder that is caused by a recessive allele, which means that in order to express the disorder, the individual has to have two recessive alleles. They need to be homozygous. And the best indicator of this is if you see a child that expresses the trait, but neither parent has the trait. So again, it's easiest to start at the youngest generation. So this is the youngest, newest generation. And in this generation, you have two siblings who express the disorder. Then you look at the parents, and they do have a parent who expresses the disorder, but then you need to check his pedigree. So you look, he has the disorder, and when you look at his parents, neither of his parents are expressing the disorder. So this indicates that this disorder is an autosomal recessive. And it is really important to check every generation, to check every individual who is expressing the disorder to make sure they have parents that do not express it. For your second experiment and your lab report, you're going to be analyzing different pedigrees. And there will be 5 pedigrees that look something like this, and you will be answering certain questions about them. The first question is the mode of inheritance. So when you look at the pedigree, you have to determine if it's X-linked, autosomal dominant, or autosomal recessive. So to analyze this pedigree, first one, X-linked, that's pretty easy to determine. You look at all the individuals who have the disorder. So all 4 of these children, these 2 parents, every individual who has the disorder. And if the vast majority are male, then it is X-linked. In this case, it's about evenly divided between males and females, so you can rule out X-linked. So your next 2 choices are autosomal dominant and autosomal recessive. So you need to start looking generation by generation at all the individuals who have the disorder. So we'll start on the right side. So here in this generation, no one has the disorder, so you move up to the next generation. This man has the generation, so you want to look at his parents. And his parents, he has at least one parent who has the disorder. So this indicates it may be a dominant. But you need to check out all individuals who have the disorder. So you can go over youngest generation to this set of 4 siblings that have the disorder. You look, and they have 2 parents who have the disorder. Then you look at this woman who has the disorder. You look at her parents, and neither parent has the disorder. You look at these 2 people, so that indicates recessive. Then you look at these 2 siblings, and you look at their parents. Neither parent has the disorder. So because you have 2 situations where you have an individual who has the disorder, but their parents don't have it, then this is autosomal recessive. So first, determine the mode of inheritance. So for this pedigree, it is autosomal recessive. Then you figure out the genotype for the disorder. Since it is autosomal, you're just using the letters by themselves. They're not on the X chromosome. And it's recessive, so it's lowercase a, lowercase a. And then you will have 3 different individuals that are labeled, and you have to figure out their genotypes. So for instance, we can say this person number 5, and we can say person number 7. I'll just give you 2. So person number 5, figuring out her genotype. So she has a normal phenotype. Since she is normal, she must have one uppercase allele because she is normal. But then to figure out what her other allele is, you have to look at her parents. And since she has one parent who was homozygous recessive, that means that her father gave her a lowercase allele. Second person to look at, person number 7. Person number 7 has the disorder, and because this is an autosomal recessive disorder, he must be homozygous recessive. The third experiment you will be doing has to do with blood typing. And blood typing refers to the 4 blood types, type O, type B, type A, and type AB. And the letters refer to the types of molecules you find on the surface of the red blood cells. A type O person has no molecules on the surface of their red blood cells. Type B has B molecules on the surface of the red blood cells. Type A has A molecules on the surface of the red blood cells, and type AB has both A and B molecules on the surface. These are the reagents you would have had in lab to test 4 different samples of blood to determine the blood type. So these are the 4 different samples of blood, person 1, person 2, person 3, person 4, and these are the reagents you would have had. So these are the 3 reagents. This would have tested for the presence of antigen A, antigen B, and then the third one is testing for the RH factor. RH factor, and the RH factor determines if your blood is a positive or negative. So when you say somebody has a positive blood, positive is referring to the RH factor, or a negative blood, it's referring to the RH factor. In order to test the samples, what you would have done is taken 1 drop of blood, so person number 1, 1 drop of blood into each of these 3 wells in a plastic plate, and then you would have taken 1 drop of reagent A and mixed it with a drop of blood in 1 well, 1 drop of reagent B into another well, and then the reagent to test for RH factor into the 3rd well. So after putting the drops of blood and the drops of reagents into the wells, this is what you would have seen. So again, a drop of blood in each well, and then a drop of A reagent in that well, B reagent in this well, and RH reagent in that well. You then would have taken a toothpick and mixed the blood around. A positive reaction is seen when you get clumping together of the red blood cells. That means that the reagent is binding to the B reagent is binding to the B molecules on the red blood cells and clumping them together. If you see no clumping, that is a negative result. So in this case, this sample of blood is positive for the B antigen. So it's positive for the B antigen, negative for A, and negative for RH factor. So negative for RH factor, that means this person has the phenotype of B negative blood. Testing more blood samples, this shows what the blood sample would look like. And when you take that blood sample and mix it with anti-A, or the A reagent, the B reagent, and the D reagent is the RH factor. So in this case, this person, you get clumping with A, no clumping with B, and clumping with D. So this is an A positive blood type. This person has no clumping with A, has clumping with B, clumping with D. This is B positive. And this person has clumping with A, clumping with B, clumping with RH factor. This person has both A and B, so they're A, B positive. And this person is negative for all 3 tests, so they are O negative. The blood typing reagents will give you the phenotype of an individual, but you will need to be able to determine the genotype for individuals. And when you are writing the genotype for blood typing, You have two sets of genes. The first is the ABO genes or ABO alleles, and for these you use the letter I. There are two dominant alleles. There's a dominant A and a dominant B. So in this case, you write a capital I with a superscript A for the A allele and a capital I and a superscript B for the B allele. So these two, they are codominant. If an individual is heterozygous, they will express both A and B alleles. There is also a recessive allele. Recessive allele is the lowercase I. So if an individual is homozygous recessive, then they are blood type O. So they're not expressing either antigen. The other part to the blood typing alleles is the Rh factor. And this is a simple dominant and recessive. So the dominant allele is the positive phenotype. The recessive allele is the negative. So this is going to involve dihybrid crosses in your lab, so two alleles. So if a person is blood type B negative, then for the B, they could be homozygous dominant, or they could be heterozygous for B with a recessive. And if they are negative, then that is the recessive phenotype, so they have to have two recessive alleles. So for a person who has the phenotype of B negative blood, they could be either homozygous dominant for B and homozygous recessive for Rh factor, or heterozygous for B and homozygous recessive for Rh factor. If you have been in lab and been able to do the blood typing on the samples, person 1, 2, 3, and 4, these are the results you would actually have seen. So use these results to determine the blood type for the 4 different people. So, of course, this is person 1, 2, 3, and 4, and these are the tests for anti-A, anti-B, and anti-D. Then, in order to do the other questions, you are doing dihybrid crosses. So remember, you first have to determine the genotype of the parents and then determine what gametes they can produce. So an example, if you had a person who is heterozygous for both ABO and Rh factor, let's say their blood type is B and they're heterozygous, so they have the dominant B allele, they have a recessive allele, they're heterozygous for Rh factor, that is their genotype. When you are determining what gametes they can produce, remember that you have independent assortment. So half of the time this dominant B will go with the dominant R. So this is one possible gamete. Half of the time the dominant B allele will go with the recessive. This is another gamete. Then the recessive I allele, half of the time it will go with the dominant R. And half of the time it will go with the recessive R. So these would be the 4 possible gametes that one parent who is heterozygous for 2 different traits would produce. The last topic to go over in your lab is transformation. And transformation is a type of biotech where bacteria can acquire new characteristics. And the way the bacteria acquires a new characteristic is by introducing a gene from another organism into the bacteria. And because the genetic code is universal, all bacteria, all living things use exactly the same genetic code. That means the bacteria are able to produce the encoded protein. For the virtual transformation, which is a computer simulation of an actual transformation, what you would have done in class, go to this website, and this is the website that you would see. So go to the website and follow the directions for going through the transformation. And in your lab report, there are a series of questions that you can answer from doing this activity.